promoter system for yeast. So promoter system of yeast, it is more complex as compared to bacteria, because when transformation with yeast was started, but most of the genes at that time, they were not expressed. And then scientists realized that there may be something different when we compare the yeast expression system to that of bacteria. And then it was realized that for yeast, 4 different type of genetic elements, they are important within the promoter system. For example, of course, there is transcription initiation point is there, but here number of consensus sequences are present that are mostly AT-rich region, and this is different when we compare it with the transcription initiation to that of bacteria. And TAT box, it is also there, but it is comparable with the Pribno box of bacteria like the E. coli. But other 2 elements, they are different. Like the 3rd element that is present upstream to the transcription initiation, and it may be called as UAS. So upstream activating sequences, या इसको सिंपली UAS कहते हैं. तो ये एक ऐसी साइट है जहाँ पे रेगुलेटरी प्रोटीन्स जो हैं वो बाइंड कर सकती हैं और टारगेट जीन के जो एक्सप्रेशन है उसको अप रेगुलेट या इनहेंस करती हैं. इसके अलावा एक और अपस्ट्रीम सीक्वेंस होता है जिसको हम URS का नाम देते हैं, या अपस्ट्रीम वी कैन से दैट रिप्रेसिंग सिस्टम और URS. इसमें अगर जीन का एक्सप्रेशन डाउन रेगुलेट करना होता है तो इसमें रेगुलेटरी प्रोटीन्स बाइंड करती हैं दैट कैन लोअर द एक्सप्रेशन ऑफ द टारगेट जीन. बट if deletions are there within the US gene, especially those that upstream and activating the expression of genes, then the expression of gene can be abolished altogether. So in addition to that, certain downstream activating sequences that are called as DAS, they are sometimes locating within certain genes that can even regulate the expression. So this diagram, it is related to the structure of typical yeast promoter. So this is the coding sequence and this is the transcription initiation point, and upstream around 120 base pair, it is the TAT box, and then some upstream activating sequences are there that can upregulate the expression of target or structural genes, and they may be present around 100 to 1400 base pair upstream. And URS, that is upstream repressing sequences, are also there that may be up to 40 base pairs upstream to the transcription initiation, and they can lower the expression of these genes. So it is you can say that the typical structure of yeast promoter. Now, promoter systems have also been developed to maximize or upregulate the expression of target genes. कि इस तरह से हम प्रमोटर को यूज करें या ऐसे प्रमोटर को हम प्रेफरेंस दें जो कि टारगेट जीन में ओवर एक्सप्रेशन को इंड्यूस करते हैं. तो इसमें स्टार्ट में जो प्रमोटर यूज किए गए वो बेसिकली वर रिलेटेड टू glycolytic pathway. Certain dehydrogenase genes were used, or some other genes that are related to phosphoglyceraldehyde pathway, they were used. So the promoter of such genes were used to overexpress the target messenger RNA. That may represent up to 5% of the total messenger RNA. So promoters initially have been used to maximize the expression of target genes. Then later on, different modifications of promoters, they were used or developed so that expression of the target gene, it may be enhanced.